present to you biochemical reactions of, in particular, for Enterobacteriaceae. So basically, if you happen to see the first slide of my presentation, you can see there Enterobacteriales. This is actually the new naming now of Enterobacteriaceae. So instead of, well, you should know, for example, for board examination purposes, that this is still Enterobacteriaceae, but with recent certification under the Clinical Laboratory and Standards Institute, it is already named as Enterobacterales or Enterobacteriales. So basically, my references, I have four. So Coleman's Atlas and Textbook Diagnostic Microbiology, and that Clinical Microbiology Procedures Handbook, and then our SOP at IARC, including a little portion of PLSI 2020 30th edition of your susceptibility testing. So the main objective of this lecture would be to explain the different characteristics and properties of bacteria, and then to describe the cultural, morphological, biochemical, and serological properties of pathogenic and non-pathogenic bacteria. To highlight this, I will be presenting to you a brief background on the principle, and then the different chemicals used, the color reactions, and some QC strains or quality control strains used to test the performance of your different biochemical reactions. Now, as a backgrounder for Enterobacteriales or bacteriales or Enterobacteriaceae, they may happen to present to you as commensal or a true pathogen. And among the different pathogenic disorder, among the different infection that may cause, may happen to be categorized generally into three, but it may happen to be also a cause of various infectious diseases in the different parts of your body. First things first, which is the most common one, would be uh, it tries now to cause enteric infections, which may happen to present what we call as true enteric bacteria. So the two true enteric bacteria include, please note of this one, Salmonella and then Shigella. But apart from that, they also include their Yersinia enterocolytica. Other than that, it could also cause now your urinary tract infection, the most common of which would be that of your Escherichia coli. And then another one is causing now infection in the respiratory tract, particularly in the lungs. You may happen to have here Klebsiella pneumoniae as one of the most common bacteria that is being isolated from respiratory specimens. But then again, being a commensal at that, you try now to note of the different considerations, before trying now to conclude that this is really the cause of infection. Okay. Number one would be the anatomic location. When I say anatomic location, for example, you have cultured a Escherichia coli in the respiratory specimens. Before reporting it in the final result, please do consider first, because Escherichia coli that may happen to be contaminating your respiratory tract may not be a true pathogen. It may happen to be like potential contamination. So not all bacteria that grows in a specific specimen may happen to be a true pathogen. So it's either it would be a commensal or that would be a potential contaminant. So please do consider in reporting later on when you will be having your microbiological report, anatomic location, and several other considerations. Now, if you have later questions, kindly note of those things, and I hope I can answer them back later. So next would be the different modes of infection with regards now to Enterobacteriaceae. The most common is food and water contamination. Again, the two true enteric bacteria would be Salmonella, particularly that of Salmonella typhi and that of Shigella. All right, and then another one would be endogenous in nature. When say endogenous, they may happen to be um, organism that may happen to be harboring your body that will try not to go to different organs. and try now to establish infection. Okay? And then another one would be abnormal host colonization. And then this is different from that of food and water contamination uh, through an insect vector, particularly that of a flea would be your Yersinia pestis. So please do consider this as a highlight of the most common pathogens that you will happen to be considering under Enterobacteriaceae. So what are the different common agents of infection? So you have lot active organisms under different tribes under your family bacteria, Enterobacteriaceae. So the one that causes now your bacteremia, UTI, bacterial pneumonia, wound infection, the most common ones are E. coli, Klebsiella pneumoniae, Enterobacter species, particularly that of Enterobacter glaucoma. Okay? Another one is Tosus mirabilis. And then for intestinal infections, we have several there. And if you happen to try to note those organisms, they belong to Salmonella and that of your Shigella. And as I have said a while back, you have your Yersinia enterocolytica. And the one that is actually being highlighted under your Escherichia coli is the 015787. This happens to be uh, highlighted in eating burgers, particularly some part in the United States may. So this would be your E. coli 015787. So for antigenic structures of your family Enterobacteriaceae, this differs among different microorganisms under this family. So most are motile, particularly they are periticus in nature. When I say periticus, they try not to line the surrounding of your microorganism. And in particular, the antigen that tries to contribute with regards to your flagella would be your H antigen. Okay? While for capsules, you have your K antigen, which you have your VI or Salmonella. Now, for cell wall, a cell envelope, you have also the cell wall. In particular, you have your Klebsiella pneumoniae having a capsule. And then for lipopolysaccharide, you have there the endotoxin, you have your O antigen, and then you have also your OLP, your outer membrane protein, and then variability in the presence of your pili, which at times is to be encoded by your plasmids. So some characteristics now of your Enterobacteriaceae would be they are facultative anaerobes, motile except for Shigella and Klebsiella, non-encapsulated, or non-capsulated or non-encapsulated except for Klebsiella, non-fastigious, non-spore forming, and then they grow on bulk-containing media. Particularly, you can isolate them making use of McConkey agar. So, as part of your diagnostic identification now for the family and Enterobacteriaceae, the presumptive identification will try now to be based on microscopy, but you will not try to identify all. You cannot identify all. You cannot identify immediately to what genus it will belong. So microscopy will just give you an idea whether this is gram negative or gram positive. And to emphasize, Enterobacteriaceae or bacteria under this family are gram negative. And they have a characteristic short, plump, gram negative bacillary or cocobacilli at times, ranging from zero point 0.5 to 2 femtometer or FM wide to 2 to 4 long. So you have here now uh, a diagrammatic representation of the cell wall of your gram-negative bacilli as compared to that of your gram-positive gram-positives. You have a thicker teichoic acid or peptidoglycan layer. Now, if you try to look closely into the cell wall now of a gram-negative bacteria, it is more complex as compared to your gram-positive microorganisms. So in there, you will be having now the different components that is actually lining now the cell wall of your gram-negative organisms. Now you have here on the other side, you have here a gram stain of a gram-negative bacilli. So this is more thicker as compared to uh, other microorganisms. So they vary between one genus to another. Some would try not to present like slender in nature. Well, the nature or the microscopic appearance of certain bacteria will also depend now on the growth components of the culture media or the environment will it grows or will the environment where this is actually being recovered. Okay, so some would try not to present you as a slender but still Klebsiella pneumoniae, or others would try not to have a more or larger or thicker now component or microscopic appearance, but still this is Klebsiella pneumoniae. But then again, it will not try now to identify a particular genus by making use just of microscopy. But this is the first step towards which you could try not to proceed with other diagnostic uh, tests or biochemical tests leading to the identification of your bacteria under the family of Enterobacteriaceae or Enterobacteriales or bacterials as you may try now to utter this one. Okay, so one important thing that you should try now to consider with regards to the identification now of the genus of a particular bacteria under the family would be, well, for hemolysis, this is variable, and you cannot try not to make use of this one, trying now to delineate or try not to zero into what particular genus a particular isolate belongs. Okay, so they may try to present you to a large, dull gray, dry, or mucoid colonies. Mucoid in particular would be mostly predilected to the genus Klebsiella. Okay, so mostly, if not all, Klebsiella isolates would have a mucoid colonies. So when you try not to look into a particular colony, when you try not to isolate or try not to culture a specimen, and it appears to be mucoid gram-negative, then you have now an idea, at least, that this belongs to the family of, or to the genus Klebsiella. Okay. Other than that, you will be having now thin film or as waves or a phenomenon known as swarming. So this swarming phenomenon actually is typical of your Proteus species. Um, I have read something on Greek mythology about the genus Proteus or the word Proteus itself. So when you say Proteus, actually you have something like a wave-like fashion changing form that will try now to present you a thin film in waves when you try now to culture an organism in the culture media. This is, well, the swarming phenomenon could be readily observed on sheep's blood agar or horse blood agar plague. Okay. Now, for differentiation now, whether this is lactose fermenter or non-lactose fermenter, you could try to have your McConkey agar as one of your agars to show whether this actually utilizes now lactose or not. So other than that, you could also have there eosin methylene blue, in which you can also observe what we call as greenish metallic sheen. So this is actually, when you say a greenish metallic sheen, this is typical of your Escherichia coli. Escherichia coli could be also a bacterial indicator of water contamination. So this is being identified when you try now to have water analysis. Okay, so with regards now to the utilization now of lactose present now in the different culture media, this will try now to produce acid, changing now the color indicator present in the culture media itself. So it will try not to present you now red colonies, meaning this is lactose fermenter. It will not present you a red colony, then that would be non-lactose fermenters. So as I had mentioned earlier, you could classify now the organisms under the family Enterobacteriaceae based on colonial appearance and reaction on your differential media. So again, as you have studied earlier, I guess in your different modules, you have their differential media that will try now to make use of different components to inhibit now different microorganisms. At the same time, the use of indicator dyes. Okay. So for lactose fermenters, example under the family of Enterobacteriaceae, we have their Escherichia coli and then Klebsiella species. Again, as I have said a while back, Klebsiella, other than a lactose fermenter, Klebsiella presents a mucoid colony appearance in the on top of culture media. And then other than that, you have their late lactose fermenters. So they will try not to still to they will try to still present you a pink colony or red colony, but on a longer incubation period. So example of which is your Shigella sonnae. And then lastly, you have non-lactose fermenters. So examples of which would be other species of Shigella other than Sone, and then Salmonella species. Okay, that includes now your Salmonella typhi causing now your typhoid fever. Now, for differential identification of your family organisms under family Enterobacteriaceae, they would be solely based on enzyme production, the different biochemical tests that you'll be using to identify the genus species level of the bacteria. Now, for screening purposes, you have there three at least before you proceed with certain panel of biochemical tests. Number one is glucose fermentation. Another one is cytochrome oxidase. And mostly all Enterobacteriaceae are cytochrome oxidase negative. And then another one is nitrate reduction. So the conversion now or the reduction of your nitrate to nitrite. So these are actually the three screening test that would try now to lead to the identification now of bacteria under the family Enterobacteriaceae. So I guess you have tried now to study this one in your biochemistry, one of which that will lead now to the production of acid, making use of the glucose as substrate. We have there your Emden-Meyerhof pathway, and in which you will be having now, again, as I've said, the glucose being now the substrate to the production of a pyruvic acid in the different pathways. You can making use of different enzymes as you go along with the pathway itself. So you have there even different acids being produced to the different pathways that will lead to the utilization or either the utilization or the what do you call this one the production of other components that may lead later on to biochemical reactions of your different bacteria. So you have also their lactic acid, you have their formic acid, acetic acid, butyric acid or the butyl alcohol, and then several others. Now, for the isolation of your family Enterobacteriaceae, you have selective differential culture media that you may happen to use. And the chemicals and compounds being used are actually variable depending now on which target you will be having. But mostly, you will be having these different components added into one um, culture media. So that includes your protein hydrolysates. That includes now, this will be trying now to provide you carbon and nitrogen source for the microorganism. And then you have also their carbohydrates being the source of carbon. And at times it becomes now a substrate. For example, you have carbohydrates like lactose, sucrose, or even glucose in one culture media that's to be utilized by the organism. And other enrichments as well could be added, okay, depending now on the type of organism that you are targeting under the family. And then inhibitors as well, so that the undesirable bacteria will be prevented from growing on the culture media, so that your target Enterobacteriaceae will be the one solely growing on the culture media. The examples of which would include now dyes, neutral dyes in McConkey agar, also heavy metals, other chemicals, and even other other culture media will try now to incorporate also antibiotics. The most common one, I guess, is you have here their vancomycin that will try now to kill the um, gram-positive bacteria, allowing now the growth of your gram-negative bacteria. Others include also pH indicators or dyes, even, and then others may include their H2S indicators. For example, in your Salmonella Shigella agar, it includes there an H2S indicator as well as your triple sugar iron agar. It includes also a H2S indicator, including also your sulfide indole motility agar. They have your H2S indicator. So those are the main components that may happen to play around is a certain differential selective media for the family Enterobacteriaceae. So the different culture media that you may happen to use in the isolation of Enterobacteriaceae would be including enrichment broths, particularly if the dosage now of the bacteria in a particular specimen is low, expectedly low, then you try not to enhance them first before you try not to plant them in various selective differential media. Okay, that includes your selenite F, or others may have gram-negative broth. Or at times, if you do not allow selectivity, then you may happen to make use of tryptic soy broth, or you have your nutrient broth, could be possible. But then again, they are not as selective as that of your selenite F. Okay, so for selected differential media that's commonly used now for the isolation of your Enterobacteriaceae, you have your McConkey agar. This is the most common one, and then eosin methylene blue. Well, we use this one as a um, part of the as one of the agar for the confirmatory test of E. coli when we try now to analyze water, drinking water here in our facility. So other than that, we have highly selective media, particularly for gastrointestinal specimen. So an example of which would be, well, mostly these three are used now for the isolation of Salmonella and Shigella agar. The most common one is your SSA, Salmonella Shigella agar, also with Hectoin enteric agar. With Hectoin enteric agar, as far as I can remember, it has also an H2S indicator, if I happen to be correct. Now, another is your xylose lysine deoxycholate agar. So these three may happen to be highly selective with a particular genus of organism. These two organisms being your two true enteric organisms, Salmonella and then Shigella. Well, to include your Yersinia enterocolytica. Now, for the metabolic characteristics of your Enterobacteriaceae, you have their number one is carbohydrate utilization. Okay, the use now of different substrates with different types of sugars. Okay, that includes, of course, your glucose, lactose, sucrose, and several others. Another is your orthonitrophenyl beta D galactopyranoside or ONPG activity, indole production, methyl red, Vos-Poscauer, citrate utilization, urease production, decarboxylation of lysine, ornithine, and arginine, phenylalanine deaminase, H2S production, and then motility. So we'll try not to go to the different biochemical tests being utilized for in the identification now of your Enterobacteriaceae. So the first one being a screening test would be your oxidase test. So the principle is in the presence of oxygen, the bacteria will try now to utilize or try now to convert making use of the enzyme phenylalanine reagent as the electron acceptor to produce an indole phenol, which is colored purple. Okay, so you have there a visual presentation of which one is positive and negative. This is very, very useful now to identify that this actually could be the particular organism could belong under the family of Enterobacteriaceae. So it could be a very, very important thing or preliminary step in trying now to identify Enterobacteriaceae. So the active component of the reagent, COVAX reagent, is NNNN tetramethyl paraphenyldiamine dihydrochloride. Okay, and then you have two methods under oxidase test. You have the filter paper method. You try now to have to streak or try not to smear a colony on the filter paper. Wet this one with saline or wet with saline first and then smear with a colony of the microorganism and then add Kovacs reagent. If the colony tries or the paper turns purple, then that would be positive. If it is not turning into purple or this is colorless still, then that would be negative. And you have a direct method as well, the plate method. Okay, but of course with plate method, this is not so advisable as compared to filter paper method. Well, currently with different innovations in their and the different products for identification making use of oxidase, you have commercial discs already that you may happen to use for oxidase testing. Okay, you have the disc is actually similar to that of the antibiotic discs being used for susceptibility testing. Now, to test whether your uh, test reagent is acceptable or performing well before trying now to use it for the identification of your microorganism, you need some QC strains for you to test really if this is oxidase positive or oxidase negative. So two QC strains you may happen to use, which is standard at times. You make use of PAE or what we, this is a code actually for Pseudomonas aeruginosa. So this is oxidase positive. And then ECO or E. coli, this is oxidase negative. So E. coli actually is a representative of your Enterobacteriaceae. So these are the two QC strains usually used now to test the performance of your reagent. So other than that, after trying now to have microscopy, then oxidase testing, then if you have a gram-negative bacilli, oxidase negative, then you may proceed to further biochemical testing. And the first one that you happen to incorporate in your panel of testing is your carbohydrate utilization. So for this one, the main principle of this is you happen to have there a substrate glucose that when utilized by the organism or the bacteria, it tries now to produce an acid. Okay? But then again, with acid production, you may entail also gas production. But you cannot try now to see gas production by merely having it as in a solution or in a cultural broth. So you have there an inverted smaller tube inside the test tube. You have, this is what we call as your Durham tubes. So that you can try now to capture really. Bye-bye. So you can try now to identify or try now to detect whether there is gas production by trying now to look at a space inside the inverted tube. Okay. So if you happen to look at the particular slide you are seeing right now, you have there, it has also an indicator dye. Particularly, this is uh, phenol red. Okay, the most common one that is being used for this type of reactions would be phenol red. So once it turns yellow, then there is acid production. Now, if you have now a gas underneath or in the inside the inverted tube, a Durham tube, then you have gas production. So you happen to read those both with acid production and gas production. So the red one there is uninoculated. So there is no bacteria that's being placed or no inoculum or no isolate was placed in such particular tube. So this may happen to be used as a control with your various reaction. So the first tube on your left would be acid production with gas production, while the middle tube would be acid production only without gas production. So these are common among species under Klebsiella, Enterobacter, Hafnia, and Serratia. So the acid being produced there, in particular among the family genus Shigella, if you happen to have a formic acid in a deficiency in the production of your formic acid, and you have a deficiency in the enzyme, well, you try na not to have their production of gas. Okay, so this is typical of the middle tube reaction. So other than that, the most common one being utilized right now in the laboratory is your KIA or Krigler ion agar, where this is the um, earlier agar being utilized under this, and then under this type of biochemical reaction. And then you have also your triple sugar iron agar. So they vary only with one particular uh, type of sugar. So both contains glucose and then lactose, while the other one contains another sugar. So that would be, I guess, one of your assignment, but actually it's already indicated in the PowerPoint presentation. So again, what's common right now that's being utilized in the lab, particularly for diagnostic purposes, is the TSIA or the TSI, or triple sugar iron agar. So how does the principle of TSIA uh, summarize? So you have there a tube labeled as A. So you have there, so this you have here an agar that has now a slant and a butt. So make sure that when you prepare a TSIA, that the size of the butt should be equal to the size of the slant. Okay, so for aeration purposes or for there is oxidation purposes, it usually happens now in the slant area. While for the absence or anaerobiasis will try now to happen now in the butt portion. Okay, so if you will for TSIA, you have also there an indicator dye that makes it color red, phenol red. So what happens there is having now your different components of the media as presented a while back. And then of course it has also H2S indicator. What happens is in the presence of the oxygen in this land, and then if there is no fermentation of the sugars present now in the culture media, then you have a reaction that is alkaline, having now a pH of 7.4, and then alkaline dip or alkaline slant, alkaline but or dip, it's the same. Okay, so that would be K over K. So alkaline slant, there's no carbohydrate fermentation either in the presence of oxygen or either in the absence of oxygen. So examples of this type of organism does not belong under the family of Enterobacteriaceae. So they do not belong. So an example of this is your non-lactose fermenters. You have your Pseudomonas aeruginosa, which is oxidase positive. So these are not actually Enterobacteriaceae. Other than that, you have also their glucose fermentation, okay? And then lactose or sucrose for TSI medium, not fermented. So this is a characteristic of NLF or non-lactose fermenters. Examples are Shigella species. Okay, so you have their mixed acid fermentation and in the butt portion. So another one would be letter B again, the bottom tube. You have their alkaline slant and an acid butt or black dip. Okay, so the presence now of black precipitate is your H2S production. So glucose is fermented. Well, glucose is always fermented. Try now to produce acid. So again, as I had presented a while back, when glucose is fermented and there is an indicator, it will try now to change now the pH, the indicator dye, pH indicator dye color change from red to yellow. So when you will be having a glucose fermentation, there would still be production now. They will be changed now in the color from red to yellow. But in here, there's a characteristic. This is another characteristic of an unlactose fermenting bacteria, wherein you happen not to have their utilization now of your either your lactose or your sucrose. These are typical under the family of the genus of Salmonella, Citrobacter, and then Proteus species. Okay, for Proteus species, they produce strong H2S or H2S precipitates. Okay, those are the organisms that usually are swarming. Okay, Proteus species. And then for Citrobacter as well, they produce, but some do not produce H2S. And then Salmonella, well, the production of H2S depends. For Typhi, you have only a lesser precipitate diffusing now in the strict area of your media. Okay, and then the last is acid slant and acid dip or acid bath. So the whole preparation, the slant and the bath portion is actually yellow. Okay, so formerly, if you go back with acid slant and acid dip or bath, you have red slant and then a yellow bath or dip. Okay, and then for acid slant, acid dip, then the whole agar is actually yellow in color. So this they try not to utilize glucose, lactose at the same time, sucrose being the source of your carbon in the system or test system. So these are typical of E. coli, Klebsiella, and Enterobacter. So to summarize again the color reactions, so for A over A or K over K, should we say? So the whole tube should be red. For K over A, red over yellow. And then the second variant is red over yellow with black precipitate. And then the last one is yellow over yellow. So those are the different reactions under your TSIA or TSI reactions. Okay. So these are examples, diagrammatic representation of your TSIA reactions. So you have there A over A. If there is a crack or bubble inside your TSIA after the incubation period, then that may happen to be production of gas. So in TSIA or in KIA, you could also try not to observe production of gas. Cracks, spaces in the agar, so those would indicate gas production. So in the slide, actually, there is one that's wrong. 1, 2, 3, 4, 5. The fifth tube is wrong, actually. I happen not to have a reaction having read from various books. So I'm sorry for this visual presentation, but that's supposed to be wrong. It's like the notion. Why did I tell you that this is wrong? It's like having now rains. When you have rain, actually in a mountain, all waters go down. So if there's acid production in this particular tube, then acid may happen to fall into the bath portion, making it yellow also. But this would never happen. Okay, that's why I told you this is wrong. I'm sorry for this visuals. Okay, so these are actual TSI reactions, uninoculated, which serves as a control. In the diagnostic lab, we usually add uninoculated media, being the control of your test reaction. Okay, so the second tube is A over A with gas. You see the cracks at the bottom or the deep portion of your or the butt portion of your TSIA, and then example of which is your E. coli. So another is your Shigella sonnae. You have their K over A. No gas production, no H2S production. And then K over K, typical of your Pseudomonas, but that this is not under the family of Enterobacteriaceae. And then the last tube you have there, K over A with gas, you have cracks, and then you have also black precipitate H2S. So K over A, G for gas, plus for H2S. So that is how you write it in your working worksheets later on. But it depends on how you try now to interpret or try now to write your presentation later on, or try now to write your reactions later on in your worksheets for identification. Well, it depends on the lab, but this is how we try now. This is how we usually write the reactions. So another visual presentation of your TSI reactions. Again, we have a control. But what is wrong with this particular tube preparation is that, as I have said a while back, the butt and the slant should be equal. If the butt is 1 inch deep, then the slant should also be 1 inch for you to have now a proper reaction to happen in the particular tube. So this happens to be a wrong preparation. So how you try not to prepare properly culture media so that the reaction will be met properly. Okay, so you have K over A for the second tube with trace or very scanty production of black precipitate. This is typical of your Salmonella typhi. Okay, and then for others, like for example, you have your K over A with gas production, and you have large precipitate, increased production of H2S precipitate. Example of non-typhoidal salmonella, well, that would be other salmonella species other than Salmonella typhi. And then the fourth tube, you have K over A reaction for Shigella flexneri. So this is an example of this one. So no lactose or sucrose fermented, and then glucose fermented. For your reading, I will not try now to tell you the ratio of the sugars for TSIA, but for your reading, I guess, try to discover what is the ratio of glucose to sucrose and then lactose present now in TSIA. And also as part of your reading, try now to read on what particular H2S indicator is being utilized or being incorporated in triple sugar iron agar together with Krigler's iron agar. So this would be very, very important later on if they may happen to be appearing in your board examinations. Okay. And then for the fifth tube, you have A over A. So glucose and lactose or sucrose is fermented with gas production. So typically for E. coli, you have strong gas productions. Okay. And then for the last, again, there's no fermentation. If you try not to look at it, almost similar with the uninoculated tube, then that would not be an example of bacteria under Enterobacteriaceae. So other than that, you may happen to have their acetate utilization. So this is used now to determine if the organism can use acetate being the substrate as the source of carbon. Okay, so you have there an alkaline blue because of the growth of the organism for positive, and then negative for no change in color being the green one. So again, in a test system, as I opt, try now to reiterate always, you try now to include always there a positive and negative control strain. At the same time, an uninoculated culture media for you to test really if your biochemical test format is working properly. Okay, so again, with this type of reaction, the presence of an indicator, pH indicator, will try now to lead to the what do you call this one? Lead to the identification or try now to lead to the visualization of a positive reaction, the change of color from green to blue. So other than that, you have also your methyl red test. This is actually trying now to note if there is really mixed acid fermentation. But again, may I remind you that we do not usually make use of this one already in diagnostic labs. But for academic purposes, we need to present this one, or this is being performed in the laboratory as part of your IMBIC reactions. Well, to note, in advance what is IMVIC. IMVIC denotes for indole production, M for methyl red test, V for Bogues-Proskauer test, and then C for um, citrate test. So I haven't not presented the rest yet. You have the first one from the in the Invic reaction as methyl red test. So this is mixed acid fermentation. Usually you try not to produce acid from a simple sugar. Commonly that you have there your dextrose or glucose. And the change of color from red to yellow is actually um, due to the strong acid that's being produced. And the pH usually of your metal red acid breakpoint, you should remember this one, that the acid breakpoint of your metal red is 4.4. Once it surpasses this one, due to the increased acidity now of the solution, then the red color will change the yellow color, what do you call this one? When you're trying to incorporate the indicator dye reagent, it will try not to change the color, making it red. That's why metal red test. Okay? So originally, the test format is for 48 to 72 hours, but with the modification of Barry et al., it can already be tested for 18 to 24 hours. Okay? So a positive reaction will try now to denote there a red color, which actually is the same as that of the original color of your methyl red. Okay. So how to perform this one is 0.5 ml aliquot of broth culture. You add there 1 to 2 drops of methyl red reagent. Then another for imbic reaction is your Vos-Proskauer. So this is, so if you have mixed acid fermentation, actually you have also mixed acid fermentation making use of this particular test. Well, in a test system making use of your, you have what we call as methyl red Vos-Proskauer medium, or what we call as MRBP medium. What we do with these two tests actually is, when you try not to, it's either you try not to inoculate one broth for 18 to 24 hours, and then after incubation, you divide the tube, the broth culture tube, and then test it separately with methyl red and then BP test. Or you could actually inoculate two separate MRBP medium, one for metal red test and one for BP testing. So either way, it could be effective. Okay, but then again, make sure that you have the same organism when two separate broths are being inoculated. So this is now for the BP test as compared to mixed acid fermentation for methyl red. This is now based on the conversion of your acetyl methyl carbinol, or commonly this is your acetylene to diacetyl. Okay, so this is due to the action now of potassium hydroxide and the presence of oxygen. So what happens now, if you will be having, if you will be adding now your alpha naphtol reagent with creatine, then it forms now a red complex. So what are common organisms that are actually positive for BP test? You have there, well, this is Enterobacter aerogenes, but currently this is already known as Klebsiella aerogenes. Okay, and then another one for E. coli, you have their negative. So you have also another control. Again, they include always a control with that of your test system. For BP testing, you can actually make use of Klebsiella aerogenes being a control organism when you're trying to have several panels of biochemical tests for you to note again if the test system really works. And then part of your imbic reaction, you have their indole production. So the substrate is tryptophan, the products being detected is indole, in the presence of pyruvic acid and ammonia. So you have two reagents having non-active component, which is your paradimethyl aminobenzaldehyde. You have your Ehrlich's or Kovacs reagent. Positive color reaction would be red. Now, for indole testing, you may happen to include there, again, either E. coli or Klebsiella aerogenes being your QC strains. Okay? So note in the slide presentation there's extraction procedure that's being part of the test system, particularly if you make use of COVAX reagent. Okay, what we use in the lab actually is Ehrlich's reagent. That's why we need not to have now extraction procedure with this. Okay, so for extraction compounds you may happen to use, you have their xylene or chloroform, but commonly we make use of xylene other than as compared to chloroform. But then again, uh, it's better to make use of Ehrlich's rather than that of Kovacs. So this is now what happens with your indole production. You have a diagrammatic representation there. And then you have here now a visual of your reaction. So if I happen to be correct, what you are seeing is a sulfide indole multimetamedium. This is actually a but preparation of your SIM agar. Okay, so red ring is positive, then no change in color, or you have now there no red color being seen, then that would be, or being visualized, then that would be indole negative. So other than your sulfide indole motility agar, you can make use of your motility indole ornithine agar, or you can make use of broth as well. Okay, broth with the incorporation of tryptophan being the substrate. So if you happen not to make use of the tube method, then you could use now a rapid spot indole method. So this is being performed by preparing now 5% para-dimethylaminobenzylaldehyde or 1% para-dimethylaminocinnamaldehyde in 10% volume over volume, making use of concentrated hydrochloric acid. Okay. But again, for rapid spot indole, there's a note there that you cannot use this one for anaerobic organism. But this is useful for aerobic organisms, facultative anaerobes like your Enterobacteriaceae. Now for tube method, you have there a procedure. This is under your clinical microbiology procedures. You have their COVAX for Enterobacteriaceae. So you have their other agars I have presented a while back as well. Okay, so this is just a preparation of the reagent. So this is for Ehrlich's. So for COVAX, you may happen to make use of isobutyl or isoamyl alcohol. But for Ehrlich's, you make use of ethyl alcohol. And then you need an extraction component that of your xylene as well. May I just clarify? I think I have interchanged what I've been telling you a while back. So yeah, it is better to make use of that of COVAX rather than that of Ehrlich's, since Ehrlich's makes use of extraction procedure. I'm sorry. So it's the other way around. Okay, so I'm sorry for that. All right, so other than that, to complete now your INVIC reactions, you have for I, indole, methyl red, Voges Proskauer, or the BP test, you have your citrate utilization. So for sodium citrate is now the substrate here being the sole source of carbon, and then you make use of co-source medium. This is the earlier preparation now of the medium for citrate utilization, but what's common right now is your Simon citrate agar. Okay, and the indicator being used is bromothymol blue. So you have there the reaction of your nitrogen component from ammonium phosphate in the medium releasing now ammonia. And again, ammonia is alkaline, changing now the medium from green to Prussian blue or blue, simple as blue. So examples of QC material for citrate utilization would be so for citrate utilization, you happen to make use of citrate. Okay, for citrate utilization, you can make use of Klebsiella pneumoniae being positive, and then E. coli being negative for citrate utilization. Okay, so Klebsiella pneumoniae positive, and then E. coli negative. Another one is urease test or urea utilization. So the substrate here is urea again, and you make use of Stewart's urea broth or Christensen's urea agar. What we make use here is your Christensen's urea agar. Positive color reaction is pink. There would be alkalinity during the phase of the utilization now of urea with urease being produced by the bacteria. So you have there a pink color reaction. For urea, you make use again of Klebsiella pneumoniae being positive, pink color reaction, and then E. coli negative for urease production. Now another well, this is not much common for Enterobacteriaceae, but you can make use also of amino acid decarboxylation. This is more commonly used for non-fermenters as compared to your Enterobacteriaceae. But may present you as well because, yeah. So for the culture medium being used here is molar decarboxylase. Now the different substrates may vary depending on the addition of a particular amino acid. So for lysine, if this will be converted, then you may produce now cadaverine or you will produce cadaverine. And then for ornithine, you have putrescine, and then for arginine, you will be having citrulline. Now the extraction compound would be chloroform with the addition now of ninhydrin reagent and the presence of bromcresol purple, there would be now acidification now of the solution, making it now positive. So usually what happens here is that from purple, it tries now to produce, you will be having now a purple color change or no change in the medium. So no change in the medium is positive, purple, and then negative color from purple to yellow, that would be negative. So this is different to that of your TSIA or carbohydrate utilization, wherein the indication of yellow may happen to be positive. While this one, yellow color reaction with addition of ninhydrin reagent would be negative. What's positive is no change in color. Okay. So for lysine decarboxylase, you have for Citrobacter species versus Salmonella species, which is 98% positive for lysine decarboxylation. So the production now of cadaverine, making use now of lysine as the substrate. Now when you say 0% versus that of 98%, take note there of the 98% for Salmonella. What it means is that not all Salmonella species are positive with lysine decarboxylation. So there is a chance that 2% of Salmonella species is negative with lysine decarboxylation. So that would the number, the percentage there, that's the meaning of the percentage present in close parentheses besides your Salmonella species there. So what it means further is that you need to have additional panel of tests if you need to further verify the species level of a particular bacteria under a specific genus. Okay. Other than that, you could have there also ornithine decarboxylation. You have for Shigella sonniae or Sonny, you have there more than 98%. Arbitrarily, I don't know why they have placed more than 98%. What, regardless of that, again, there's a chance that this would also entail that some would be negative as well. Okay, while for Shigella boydii, you have 2.5% positivity. And then for Klebsiella species, most of them are negative. And Enterobacter species, most of them are positive. Okay, so you are building already a panel of tests from the earlier presentation to identify a specific species under the family of your Enterobacteriaceae. So for the carboxylase dihydrolase test, you have for enteric gram-negative rods well, these are not actually under your Enterobacteriaceae, could also be used as a panel of tests. So the most common with having now a positive uh, test with this would be spreading indole negative proteus species. Okay, so the rest I just highlighted them. Mostly are, well, the other, this is again, this amino acid decarboxylation test is more useful for non-lactose fermenting microorganisms, oxidase positive. Then another test is your phenylalanine deamination or deaminase production. So this is actually useful among the PMP group of bacteria. PMP denotes for Proteus, Morganella, and Providentia. So the sole source of carbon here, the source of carbon and nitrogen here would be yeast extract, and then the reagent is 10% perichloride. So what you do is you try now to have their strict on the slant preparation of your media, and then after incubation you try now to flood now the slant with 10% perichloride, and then you have green reaction or change in color in the slant area or on the colonies, then that would be positive for deamination, phenylalanine deamination. So phenylalanine is actually another amino acid. So take note with the different enzymes all throughout the presentation, the absence of the enzyme will entail no production of the byproduct from the substrate. Okay, so for example, if you don't have phenylalanine deaminase, then there would not be, or there is no conversion now of your substrate into the byproducts, meaning there would be no reaction to the reagent being added into the test system. So again, please do note also in your review, substrate, enzyme, byproducts, at the same time the reagents plus the active component of your reagents, and then the positive color reaction. So that will be a way to better review your different biochemical reactions. So this is just a visual presentation of your decarboxylation and deamination, including your urease. Again, for urease testing, you may happen to have a slant preparation of your media, or at times you could also have broth preparations. Okay, so you have the lysine decarboxylations and ornithine decarboxylation. Again, these are negative since the colony turns the broth turns yellow, it's supposed to be purple. So this would be negative. No change in color or purple color will try out to denote positive color under decarboxylation. For phenylalanine deaminase, the positive color is green. At times they try not to call it as olive green. And then for urease testing, you have their pink color reaction if this is positive. Okay. So again, note off for lysine decarboxylation, substrate would be lysine, ornithine for ornithine, phenylalanine, you have their yeast coming now from yeast extract, and then for urease, you have your urea. So those are your substrate, and then you have the enzymes. So you have it's being tested in the system: urease, phenylalanine deaminase, ornithine decarboxylase, lysine decarboxylase. Positive color reaction: yellow negative for LDC, ODC negative yellow, positive would be purple, purple. Okay. Now for phenylalanine deaminase, positive color is green, and then for urease, positive color is pink. Uh-uh. And then another one, this is not actually, if I remember it correctly, not routinely part of discussions under the family bacteria, Enterobacteriaceae, but we have incorporated this one as one of the biochemical tests for our diagnostic purposes. So you have here, usually this is commonly used for gram-positive organism, particularly for Enterococcus faecalis. But then again, if we happen to have included this one in in our regimen or panel of tests, we happen to note that Klebsiella pneumoniae usually produces or produce bile esculin also as well. So the blackening is positive, no blackening on the medium is negative. So you can omit actually this one as part of your panel, but nevertheless, even though this is not more common among in testing now your Enterobacteriaceae, could still be an added test to determine really if that would be the genus species of your bacteria under the family of Enterobacteriaceae. So the components there, you have dark um, 40% bile, and then if the bile is being hydrolyzed to esculin, then you will be having now precipitation of a black component. So this is another agar agar preparation of your biesculin test. You could have it in plates, or you could have this also in slant preparation. But if you note of the preparation of the tube media there, you have a very high, the height of the dip is the deep portion is too high. Okay. But it's better to have now a larger portion of slant when trying to prepare your bile esculin agar. All right, so other than that, you could also have their lysine iron agar as part of your panel for biochemical testing. So for lysine iron agar, you could actually test here various components or various biochemical tests. So an example of which is the lysine decarboxylation, the amination. Now for lysine deamination decarboxylation, here you could actually predict already if there would be a particular genus of bacteria under the family of Enterobacteriaceae, making use of this agar. An example is your proteus. Once the slant area turns red, actually it denotes of a bacteria under your PMP, your Proteus morganella and providentiae species. Okay, so an example of which would be various reaction of LIA is that purple, then you have they're also purple with black precipitate, then that would be Salmonella typhimurium. Typhimurium is different from typhi. Okay, you have also the purple over yellow, so alkaline over acid. So the first one is alkaline over alkaline with H2S, the blackening. Again, for your reading purpose, for your reading, you try not to discover or try not to note of the indicator, that speech indicator being used in the media. At the same time, this one, the H2S indicator. Okay. For Shigella boydii, example of a reaction is of K over A under lysine iron agar. And then E. coli, that's no reaction of your medium, that's K over K. And then red over A, or red denotes the amination. Okay. In LIA, red denotes the amination in the presence of oxygen. Okay, so the example is Providentia stewarti. So you have there, actually those are the different control organisms that you may have in use when testing with LIA. So, well, I've been telling you most of the indicator dyes, but I have a table present here for the different H2S or indicator, H2S indicator for different media. So as I have said a while back, for triple sugar iron agar, we have ferrous sulfate, same with Krigler's iron agar, and then ferric ammonium citrate for lysine iron agar, and then the rest. Okay, so for sulfide indole motility agar, you have peptonized iron being the H2S indicator. So the main principle of this one would be the release of sulfide from cysteine or thiosulfate by the enzyme. Then it will try now to couple your sulfide, producing now their precipitate and detected by the presence of heavy metals. Okay, so this will turn out to be black precipitates. So another test would be your motility test. Okay, So you could actually try now to observe this one, making use of your limb or lysine decarboxylase indole motility agar, either your sulfide indole motility, and then you have also your, actually this is MIO, not MOI. So motility indole ornithine decarboxylase. So for the different indole testing here, you can make use of either COVAX or ERLIX. Again, COVAX doesn't present you extraction procedure as compared to ERLIX reagent.
Sir Wilson,
Yes, ma'am.
pa-interrupt naman. Pwede daw pa-expound sa decarboxylation.
Decarboxylation sa part, yung pan, ma'am, yung tatlo.
Ah,
Well, deamination happens in the presence of oxygen. Decarboxylation is usually anaerobic in nature. Okay, so as simple as that. Now, in the presence of oxygen, when making use now of LIA, then it turns now red in color. But if this will be decarboxylation, making it will try now to uh if you will, okay, here. Decarboxylation, again, the amination happens in the slant area, decarboxylation happens in the butt area. So anaerobic, aerobic. Okay. Now, if the amination is positive, then there will be change in red color from a purple to red color. While for decarboxylation, um, there should not be change in color for decarboxylation. If it turns from purple changing now to yellow color, that would be negative for decarboxylation. Okay, so you have here lysine deamination, but no decarboxylation. In the last, if you're see, if you are in the last tube, so you have the amination, negative for decarboxylation. While in here for E. coli, you have a negative deamination, but you have a positive decarboxylation. Okay? Okay na po? Okay na, ma'am? Ma'am?
Wenson. Okay, proceed tayo.
Amen. So, ganun lang po yung decarboxylation tsaka deamination. So, it happens to be the environment where it tries now to convert the substrate into trying now to present you a possible reaction. Okay? So, again, deamination happens in the presence of oxygen. Decarboxylation happens in the absence of oxygen. So, positive again for decarboxylation, no change in color, purple. While for deamination, it tries not to change the color into red. So again, for the most notable deamination reaction is among the family of PMP, Proteus, Morganella, and Providentia. Okay, so I hope I have justified it properly. If I'm wrong, Ma'am Dorina would do the rest. Di ba, Ma'am Doms?
When sir.
Sorry, Abram. Bigla ng mga request na ito. Anyway, okay. So in here, so siguro kaya siguro pina-expound naman dahil dito. Now for uh, lysine decarboxylase indole motility, the preparation actually of the media here is but kaya mapapansin nyo, if you will be having lysine decarboxylation, it happens in the butt area. There would be no change in color. Now, you could also observe here a lysine deamination, but in a minimal amount, the upper portion of the butt area. I don't have a visual actually of the tube, but you could happen to see a reaction similar to that of the LIA. Medyo may red ng onte doon sa taas ng but preparation ng medium ito if you will be having lysine decarboxylation. The same true with your MIO media. Ganun din yung labas niya. Kung merong deamination na nangyayari, usually yung siguro at around siguro mga 1.1 cm, 0.1 inch, maliit lang na portion, nagre-red kung minsan. But apparently, it cannot, it is best observed making use of LIA rather than this particular test platform. But again, focusing on this, motility lang kasi ang dinidiscuss dito. So, if you'll try now to have a stub, okay, into the butt, half stub lang, wag yung i-through and through yung stub ninyo, sa half lang. You'll try not to see precipitates around the stub area, then that would denote now motility. Others would present as cracks emerging from the stub area. So that's still the positive for motility. So those are typical reactions that may happen to observe with this type of preparations. Also, others would present like from the stub area, there's a growth, tapos parang may cloud of black precipitate, that's still motile. So those are different reactions that you may happen to see making use of this type of media. Particularly, yung sinasabi kong may black is yung SIM. Okay? So stub, half stub mo siya, tapos doon merong kang kikit. Pag produce yan, merong actually parang cloud of black precipitate, motile pa din yun. So those are examples of reaction you can observe under this type of medium. Okay, all right. To summarize pala yung mga streaking procedures pala hindi ko na i-present. Pag pag Krigler iron agar, LIA, yung mga Krigler iron agar at LIA, usually you try now to have a stub which is talagang through and through doon sa bottom ng tube and then streak doon sa slant area. Yun ang pag-streak nyo doon yun ang gagawin nyo sa TSIA, Krigler Iron Agar, tsaka LIA. Lahat naman ng slant, usually, you streak them. Okay, doon sa surface ng slant portion. And then for broad medium, usually, you need to tilt the tube, try now to inoculate the organism into the space from the space created by trying now to tilt the tube with that of the broth, and then try not to return it back to your normal position. That's how to inoculate a broth culture medium. Okay, so those are the techniques that you may happen to do. But this is best appreciated really if you have it done in the laboratory, not just by merely trying now to tell you or even trying now. Well, I hope it could be demonstrated on visually online, but I guess this is also impossible with this type of presentation. Okay, so for clarifications, if I happen to have missed or I happen to have mispresented, Ma'am Dorina would try not to correct it for me. Okay, thank you. All right, so motility in dull medium. This is the one I'm telling you a while back. Meron pa akong visuals nito. So you try not to have here there's wrong inoculation here. Supposed to be this is just a half stub, but if you happen to have seen the streaking technique or the stubbing technique is medyo magalaw-gaw siguro yung kamay ng nag-streak, so dapat straight lang talaga para at least kasi kung hindi mo siya in-streak ng diretsyo na ganun tapos naigalaw mo na slide na ganun, baka ma-mistaken mo later on as motile yung organism dahil na-spread mo doon, hindi solid na stub yung ginagawa mo. What I do is yung parang abrupt na stub tapos iangat mo kaagad yung needle, inoculating needle. So that is what I do. But again, this is best done when trying out to have it in the laboratory. So the first tube for motile indole ornithine decarboxylation procedure, you have there uninoculated, so purple in color, indole negative, so the addition of either Covax or Ehrlich's reagent. But then again, if you may happen to make use of extraction compounds before you add the reagent, that you might do so depending now on what reagent you'll be using. So indole negative, the flexneri, positive, the third tube for E. coli. And then you have there, the fourth tube is for motile. You have been, I hope you can see it clearly there. You have their precipitation in the stub area. Okay, and then there would be purple color Okay, so that would be positive for ornithine decarboxylation. And then that would be example of Salmonella, Cerobar, Newport, although this is not common really. Okay, then you have also their non-motile ornithine negative, the one, two, three, four, the fifth tube from your right, from your left. And then non-motile, the same time ornithine positive, a purple color change for Shigella shonmi. So this would be making use of your motility indole ornithine agar. And then last, but I guess this is also not so common with the testing now your Enterobacteriaceae, but happens to be being incorporated as well in our panel of tests here. But we'll be having here this test useful for Serratia species because these species are usually positive for DNA tests. You have here on your left, you have there a stick of microorganism, and then on your right, you have also another one. So if you happen to the the zone of clearing around the colonies, that would denote a positive reaction. So you need to add after incubation, you need to add or flood now the colonies with one normal hydrochloric acid. Now, if there is clearing positive, no clearing, that would be negative. Again, this is usually positive among species of Serratia. Okay. Other tests include pigment production. Okay. So, well, what did why did I tell you that one? How to do the pigment production is just as strict now as slant of nutrient agar or tryptic soy agar with the colony of the organism, incubate it for 24 hours, and look for pigment production. Usually, Serratia presents you a red pigment, but not most of the time. Okay, so again, the panel of tests will try now to vary depending now on type of organism or targeting for a particular genus species. Okay, so okay, for negatives, you have their Klebsiella and then Enterobacter. So this is a diagram on how to work up. This is actually taken from the clinical microbiology procedures. A gram-negative bacteria that grows on BAP and MAC, okay, blood agar plate and McConkey agar. So on blood agar plate, there's no swarming. Perform oxidase testing. If this is oxidase negative, then you try now to proceed with further but various testing that you may happen to have. But again, this is a complex one as compared to what we are using. We have a simplified one. But this is useful when you're trying to zero in a particular species as well. Okay. But if you try not to note there, you have MUG that's specific for E. coli. Okay. Mhm. Okay, so I have here another diagram, I guess. Okay. So anyway, I hope I didn't miss other tests. Anyway, these are common pathogens and rapid conventional methods with a suspected colony on your blood agar plates or McConkey agar. For example, you have their E. coli, oxidase negative, indole positive, and usually hemolytic. So that would give you an idea that this is E. coli already without testing the whole panel of tests. I will trying now to evaluate the whole panel of biochemical tests that you have performed. Take note that not all the biochemical tests are very useful. An algorithm would try now to zero in immediately a specific genus species. That would be very useful. I'll be presenting you later on an example. Now for proteus, as I have said a while back, if there is swarming phenomenon on the blood agar plate, then that would already give you an idea that this belongs now to the genus Proteus. Okay, so you have their indole negative swarming, Proteus mirabilis, Proteus vulgaris. You have also their indole positive. So that's the difference between the two types of Proteus with swarming phenomenon. So this is an example though. So this is what we are using in the laboratory for diagnostic purposes. So you have there You classify now the organism that grows on Maconchi agar, whether this is lactose fermenter or non-lactose fermenter or late lactose fermenter. So if this is late lactose fermenter and you make use of SIM, sulfide indole motility, it happens to be positive, then pwede itong Citrobacter. So you could go directly now to the table for Citrobacter for the speciation of specific for speciation now of the organism. Now, if this is negative, you check further the indole. Is it positive or negative? Now, this is negative, then this is the different genus for that particular reaction. You have Hafnia, Enterobacter, or Klebsiella. But if this is positive, try now to check the citrate. If this is citrate negative, then more likely this is E. coli. So if you try now to go back to the upper portion now of the algorithm, if this is H2S negative, indole positive, citrate negative, then that would be E. coli. Okay? So that's the simplest example that I could give you. But then again, as I have said a while back, chances are there are some species of organisms that may happen to present positive, strong positive, or 100% positive reaction with a certain biochemical test. That's I told you a while back. Now, if this is H2S positive, you can try now to zero in or try now to focus on Citrobacter. But then again, there are also H2S negative, indole positive, citrate positive with LDC or lysine decarboxylation negative that may happen to be Citrobacter also. Okay? So this would be very, very helpful in trying now to identify a specific uh, genus species under the family of Enterobacteriaceae. Okay? So that would be for non-lactose fermenter. Now for non-lactose lactose fermenters. Now, for non-lactose fermentation, then phenylalanine deamination would be useful if this is positive, and then H2S positive as well. Then, of course, you have swarming. Then this could be produce. So why am I presenting you this one? This is useful to just go with the genus of your specific microorganism. Now, if you have already zero into the genus, then you could go to the different tables now of your different organisms. Okay, so if going back now to lactose fermenter, H2S positive Citrobacter, then go to table 1. This is what we are using. So try not to take note of this one. That's why I told you that not all organisms are citrate. I mean, present strong positivity with certain biochemical tests. Take note of Citrobacter frundi, 78% positive with H2S, but not all are positive. So the remaining percentage is negative. So this is a combination of your panel of tests for you to identify this specific organisms, genus species of the organism. So for Citrobacter, you have this panel that you may happen to use. Okay, this is lifted actually from Kohneman, at the same time from the book Manual of Microbiology book. Okay, we have combined this one together with our ITM, the book of the manual of RITM. So we have combined those things and used it in our laboratory. So other than that, you have also Edward Shella species. You have their 100% are actually positive for H2S for Edward Shella tarda. And then you have the rest of the biochemical test and several others. Now, may just well out emphasize, formerly Chronobacter sakazaki was Enterobacter sakazaki. So you might as well try now to read on because some of the genus of the species, a genus of your microorganism are changing over time. This may happen to be due to molecular techniques that's being implemented or being utilized for research purposes. That's why there are changes now in different names of your microorganisms. Okay, for this one, let me just highlight here your E. coli. There are two um, well common E. coli that we are looking in when we try now to test here at the laboratory. You have your E. coli, the common E. coli, and the inactive E. coli. So for this one, we try now to look into further that some of the inactive E. coli may happen to be non-lactose fermenters at some point. But nevertheless, we try to consider other biochemical tests. Like, for example, you have your arginine dihydrolase, you have your ADH there on your right area. 3% lang ang kanyang um, positivity rate. So you'll be having 97% that's negative. So you select a proper biochemical panel that will try now to lead to the identification of your E. coli. So for motility, you try not to look at it. The most common E. coli that you'll be having is 95%, medyo yung iba hindi din motil. So yung inactive naman, medyo 5% lang. Most of the time, hindi na siya motil. So those are considerations really of how to use your different biochemical panels. Ma'am, sorry, wait lang po. Doc. Yes, Doc. Yep. Ay, hindi ko alam, Doc. Wala. Oo. Wala. Hindi ko namin nakita, Doc. Oo. Well, hindi, Doc. Well, wala ko no. Sige, 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 Doc. Well, sige, Doc. Ma'am, sorry. Timawag lang ni Doc. Okay, so other than that, moving on with your Klebsiella species, as I've said a while back, for colony morphology, they are usually mucoid. Okay, but there are some colonies of Klebsiella also that are not mucoid in presentation when they grow now on your agars, particularly on your Makonki agar, but most often than not, they are actually mucoid in nature. So to note of, you have also their changes in the name of your Klebsiella orthonilitica. So this is your Rotella already, Rotella orthonilitica, but still under this genus. Just the same, they have changed the genus of the species, but it belongs to this group. Okay? So you have their H2S negative, indole negative, motility negative, and then the rest. And if you are like seeing the different tables, the different panel actually changes. Okay, so that depends on how better to identify the genus species level, making use of the specific biochemical panels. You could actually test all the different panels, but when you try not to zero in to specific genus species, you just consider this different panel of test results. Okay, you need not to read over all those different biochemical panels. You can test them all, but select only those that are present in here for you to identify the genus species. Okay. Um, just a good note perhaps to consider when you're trying now to differentiate Klebsiella pneumoniae and Oxytoca. Well, these are particularly for Oxytoca. This is a common nosocomial infection, particularly in neonatal intensive care units. How we try now to differentiate this one? We try now to look at the indole test. Okay, most often than not, indole is positive for Oxytoca as compared to pneumonia. Okay, so that's the way how we try now to differentiate this one. If both are mucoid and all reactions are almost the same, then we zero in with indole only. Okay, so I hope you get the idea how to use this table. And then you have your PMP group, as I have said, swarming phenomenon is one of the considerations that you will be trying to note when you try to zero in with this type of bacteria. Okay. Let me just go back. Okay. All right. So those things that you need to consider. Okay, for Salmonella, again, the more common or important organism here is typhi. Uh-huh. Usually you have minimal precipitation of H2S in the stub area of the TSI. Okay. Okay. Then Shigella you have also there. And then for Sriracha, you have also this one. Okay. So most often than not, you try not to perform, please do consider in performing DNA testing at room temperature usually. Okay. And most of them, except for Ponticuli, they are positive with DNAs, except for a few having now variable reactions. So these are some imbic reactions common among the most common organisms I have selected. So you have here TSI that's uninoculated, several others. So these are the control panels. Now for the biochemical test of your E. coli, on your right, you have A over A gas positive, citrate negative, BP positive, motility positive. So these are there is turbidity now uh, in the stub area, and then there's no blackening, so there would be H2S negative. And then for BP, this would be negative. A positive result with BP would be red ring. And then for your LIA, this is K over K. So there is no deamination since red is the color for positive, and there is decarboxylation. There's no change in color of the bad portion. Okay, so I hope you get this one. So this is A over A. There is utilization of glucose, lactose, and sucrose in the medium, acid production, and then there's also gas production. So A over A, gas positive. Citrate cannot be utilized as a sole source of carbon, so citrate negative. Tryptophan, there is the presence now or production of tryptophanase enzyme by the bacteria, converting now your substrate, tryptophan, trying now to produce now the compound. When you add now the reagent, Covax or Ehrlich's, then you have their positive ring color. And then you have also their BP test and then LI. If you try na to denote this one, wala na kayong methyl red test. Kasi hindi siya masyadong nagagamit for differentiation of the species of the organism. So another example would be your Klebsiella pneumoniae. So here you have also gas production or A over A gas production, citrate positive, same, non-motil, indole negative as well, and then H2S negative. Enterobacter at the same time, Klebsiella species are BP positive. So you have here now a red ring on the top of your medium. Okay. And then LIA, the amination negative, decarboxylation positive. So again, the note of the decarboxylation of amino acid here, the amino acid present is lysine. So for Salmonella typhi, why did I place there a question mark? Because for Salmonella typhi, maliit lang kasi ang black talaga na produce. When I happen to get this one from the internet, that's what I happen to get this one from the internet with no validated information. That's why I have the question mark here. More likely, this is still Salmonella, but this is not typhi. That's why I placed there a question mark. Maliit lang usually ang H2S pag Salmonella typhi. Okay, I hope I made my point clear here. So if you happen to read this one, TSA reaction, you have K over K, alkaline over alkaline, plus or H2S positive. There's no gas production. And then for citrate, this is negative. A positive result is blue. Again, To review your biochemicalness, you note off properly the indicator used, positive color, and then the reagent that's being utilized for said biochemical tests. Now, for sulfide indole motility, you have there here a black protruding accumulation or precipitation here. So this is motil positive, then H2S positive as well, and then there is a red ring here. So this is indole positive. Okay, H2S positive, indole positive, motility positive. BP positive here. And then you have there no deamination. We have a purple color. And then positive decarboxylation, no change in color. All right, so this is your Salmonella cerobar Newport, typical of most non-typhoid Salmonella. You have there K over K, gas positive. This is K over A. H2S positive, gas positive, and you'll be having here, yeah, and then you'll be having here your urease test negative. You have here your LIA, you have there K over, this is K over A, H2S positive. Okay, this is citrate positive, motility motility indole ornithine. So you'll be having their positive ornithine decarboxylation. You have turbidity for indole. You have negative result there. So for paratype A, you have K over A, gas positive, urea negative. Um, LIA, you have their K over A. You have citrate negative, so you have also their indole negative for MOI, and you have their motility positive as well. Now for Shigella, you have their K over A, citrate negative, motility negative, indole negative, and H2S negative. BP test positive, and then you'll be having, sorry, you'll be having there an LIA reaction of negative deamination, negative decarboxylation. As I have said a while back, for decarboxylation, there's no change in color. So this is a typical example of negative decarboxylation. From purple, it changed into yellow. So Shigella, so Shigella Sony, you have K over A, urease negative, LIA K over A, you have citrate negative, you have also their motility, and then you have their indole negative as well. So for Flexneri, the difference between these two, we have, okay. So this is E. coli. So you have A over A, gas positive. There is strong acid uh, production. That's why you have also there an increased gas production. So you'll be having here urease negative. For LIA, you have there, it's not LIA. Okay. So they have their deamination negative, you have also decarboxylation negative, citrate negative, and you have your MOI. Well, if you try not to observe, making use of MIO is less variable as compared to making use of sulfide indole motility. You can observe better other parameters making use of SIM. That's why in the lab also we make use of SIM rather than MIO. So I think there's a lot to absorb in here, but I hope you could have better reading and apprehension later on and clarification if you might have while you try to read your books. Anyway, there are changes in the established genera of different genus of your Enterobacteriaceae. So again, as I have said a while back, there would be variation already in how we're trying to look at this different organism on the phenotypic characterization of such in the laboratory. But nevertheless, let me just present you some of which we have here. Tribe 1 of the family Escherichiae, you have E. coli. Well, this is, if you try now to have it as an enteric microorganism, you have your E. coli 0157H7. This is sorbitol positive supposed to be. That's why in the laboratory, when we try now to culture stool, okay? If this is as plain as E. coli only, that is not so significant or that's not too pathogenic. But if this is E. coli 0157H7, then this become positive if you culture it from stool. So how do we culture that one? You make use of sorbitol McConkey agar. Okay? If sorbitol mac if this is sorbitol positive, then more like or most likely that would be a serotype of 0157H7. Okay, that would be for E. coli. If E. coli was isolated from stool, significant only is sorbitol positive. If this is sorbitol negative, then that's not a true pathogen. Okay? But if E. coli is isolated from blood, then most likely that would be a true pathogen as well. So E. coli inactive, so you have their delayed lactose negative or delayed reaction or late lactose fermenter, should I say, or lactose negative E. coli strain, and usually they are non-motile. If we recall it back from the table, there are around 5% only that's positive for motility as compared to 95% with that of your ordinary E. coli. So, and then several other organisms. And if you have here under Escherichia, Shigella is also under this tribe. Okay. And several others. I guess if you happen to have, I hope Ma'am Dorina, you have a book of Coneman. Mas maganda kasi siya talagang mas kompleto as compared to Sibelian Scots. Okay, so careful also in the writing of the genus species of the organism. For type P should be capital T, not small letter T. Okay, and then several others. For Citrobacter amalonaticus, this is Liviniae amalonaticus already, something like that. So you have also for Enterobacter sakazaki, that's supposed to be Chronobacter sakazaki already, and then several others. For Citrobacter kozeri, this is Liviniae malonatica, and then Citrobacter diversus, and several others. Now here you have also their tribe Klebsiella. You have Klebsiella pneumoniae subspecies Rhinoscleromatis, etc. Um, what else? So these are the more important things I guess you need to note only. The biochemical reaction of each common microorganism under the family Enterobacteriaceae. So I guess you try now to make a tabulation of the different biochemical the organism and then their biochemical reactions. So this is a more simplified tabulation of your how you try now to present your biochemical reactions. But more profound one or summarized one was the tabulation I have presented being used in the laboratory. So again, this is a more profound biochemical test, making use of several other tests other than those mentioned a while back. Like for example, the use of other sugars like Arabitol, Celtobios, Dulcitol, Lactose, Sucrose, Mannitol, and several others. But I highlighted again E. coli and the inactive one. So, so motility, what else would zero in this one? Almost the same ang reaction nilang. Medyo nagkakatalo lang talaga is how they present their colonial morphology and their motility presentation. So, with phenotypic characterization, kailangan nyo talaga ng magandang panel para ma-identify nyo yung organism. to the genus level. You cannot say that it would be like Enteroescherichia species only. You need to speciate this one at least. Well, there's some organism you cannot speciate in for diagnostic, but for this type of organism, you should try to speciate this one. So there are several strains of E. coli, but notably, they're trying to either be hemorrhagic in nature, okay, and several others. So Shigella toxin type and several serotypes as well. So, well, again, the more common that's being presented over and time again is 0157H7. The one in black in bold are actually the more common type under each type of the diarrheogenic E. coli. Well, this is the molecular pathogenesis, already an example of approach being utilized in the discussion of the pathogenesis of a certain microorganism. But this is just in general presentation, depending on the type of toxin present in E. coli. If this is hemorrhagic, toxigenic, or diarrheogenic E. coli, and several others. So how they present themselves to cause now diarrhea. So this is how on a molecular point of view. But then again, I guess this is best discussed perhaps in your cytogenetic classes or perhaps in the pathophysiology of various microorganisms under the family Enterobacteriaceae. So I'm just presenting an overview since the highlight, I guess, of this lecture was more on the biochemical tests. So other than that, we have actually isolated one uncommon E. coli strain, strain 44, if I have it to be correct, last year or the other year, which now this is being called or this is named as Lersia adenocarboxylata. But then again, this is in general E. coli still. Okay, or Escherichia. Formerly, this is Escherichia adenocarboxylata, but now this is Leclercia adenocarboxylata. And then for virulence factor on the molecular level of your E. coli examples, you have lots actually. And the pathotype depends on the type of toxin that you will be having. So for to highlight a few, for example, you have your tox B for enterohemorrhagic, for activity and effect, you have adhesion, you have their adhesins, you have your EFA1, leaf A, long polar fimbriate, several others. So there are a lot actually if we try now to incorporate already molecular biology and several others. All right, so differentiation of E. coli from Shigella, if you happen to be confused with the biochemical panel that you happen to have had when you try now to test one isolate. So how to try now to differentiate this one or try now to exclude one isolate from the other to a specific genus, then you need to focus on this panel. But there's one that is not common, actually the musate or the musate utilization. But if you try now to look at it, motility-wise, E. coli is positive, both are negative. Okay, so D stands for variability, so it may happen to be positive or negative. So if this would be the case, you add additional biochemical panel for you to further look into what particular genus you will be having. So another is for Salmonella species. So these are actually the recommended panel that you will be having. But again, these are not usually, the others are not usually common in the lab. That's why we have had an adoption of the tabular presentation a while back that was lifted from the ARSP of our ITM. And when I happen to have referenced that one from the clinical microbiology procedures, I just added a few for us to include in a panel of biochemical tests for our institution. So you have also those things. So if you have there, kindly look at the Salmonella typhi. There is quick H2S production. That's why I told you a while back, I placed a question mark on one of the tubes since there is large order there is an increased production of H2S, which happens not to be a characteristic of your Salmonella typhi. Okay, for Enterica, well, that would be for speciation already to specific group strains already. And then this is for Pantoya, agglomerans, and other Enterobacter, Liliota, Pluribacter, and Cosaconia species. So you have also that one. But again, these are more on commensals rather than true pathogens. But again, rare infections may occur from this type of bacteria under the family of Enterobacteriaceae. More common, I guess, in our institution is your Enterobacter cloacae subspecies dissolvens. We happen to have isolate these ones already. Cloacae subspecies cloacae, cancerogenous, and erogenous, and agglomerans. The rest, we happen not to have encountered yet those organisms. And then you have here for Klebsiella and Rotella. Now, formerly Rotella orthonolitica was Klebsiella orthonolitica. You have their differentiation. And mostly, um, the common isolate would be that of pneumoniae, orthonolitica, and oxytoca. So they are differentiated further. For oxytoca to pneumoniae, the indole. Now, if you will be having now orthonolitic and oxytoca, then your ornithine decarboxylase would be the best option for you to differentiate this one. And again, they are usually mucoid. And then you have also serratia there. So the variability of the pigment depends on how you try now to culture properly the organism. Okay. And that's why you try not to look at serratia, marcescens, subspecies marcescens. You have to be there variable because at times it will not present you really the red color pigmentation. What I have observed is that when I place it under prolonged incubation at room temperature, the red colored pigment usually appears. Or when you try now to store further the isolate, the red pigment appears usually. And you will appreciate now the red pigment of your Serratia marcescens. But apparently, they present as colorless colony on uh, trypticase soy agar after 24 hours of incubation. So literally, you cannot expect that it will always present you the red colored pigment. Okay, now for Providentia, Morganella, and Proteus, you have here also variation in differentiation, making use of this type of panels. But again, adonitol is not so common, but we have maltose arabinos, but not arabitol, trehalose, and inositol present in our institution. Okay, so these are not really that common, but what I can highlight here is your Chronobacter sakazaki. Formerly, this was Enterobacter sakazaki. And then, as I have said, we have isolated also Cancerogenous and then Cloacae in our institution. So Raffinus we have, Melobius is not so common, except if these are incorporated in an automated identification system. We have automated identification system, but at times the machine cannot identify this one. That's why we resort into manual biochemical testing. And this one, you have the less common type of Enterobacteriaceae. So Erwingelia, Leclerria, as I have said a while back, adenocarboxylata, Molerella, Ranella, Tatumelia, and then we have Photorhabdus symbiotica. So these are rare, unusual cause of infections. But they may happen to cause infection, depending on the pathognomonic condition and the presentation of symptoms in the patient or to the patient. And lastly, I hope we have also Yersinia species. So you have there several others. But again, uh, we happen ha I happen not to identify yet one, but of course, epidemiology should also be considered now in the uh, finalization of identification of specific organism. Okay, the more common one is Yersinia enterocolytica with their biotypes. So you have 1A, 1B, 2, 3, and 4, and 5. So you have here the panel of tests, biochemical tests that you may happen to use for you to identify if this is of the various biotypes. And lastly, apparently I should not be presenting this one, but you may happen to identify a particular organism even at the genus level. But apparently, when you're trying now to perform susceptibility testing, it will present you these various reactions or susceptibility. R denotes persistence. Like, for example, it is almost always, if not always, resistant to ampicillin. For example, Klebsiella pneumoniae, Oxytoca, and Variocola. Dapat resistant lagi sa ampicillin. Ang ibig kong sabihin dito is, kung yung identification mo is Klebsiella pneumoniae, tapos nung nag-susceptibility test, more likely, wrong yung pagka-identify mo sa isolate mo. At hindi siya Klebsiella pneumoniae, hindi din siya Klebsiella oxytoca or either Variocola. So dapat kailangan nyo din i-consider later on, hindi lang yung biochemical reactions, kundi pwede din kasing gamitin kung minsan yung susceptibility test ng particular isolate pag ginawan mo na siya ng yun nga, AST. So this is what I'm trying now to emphasize by presenting you this one. Like for example, for E. coli, lahat pwede, susceptible siya. Wala siyang resistance. This is what we call as intrinsic resistance. Innate doon sa bacteria na resistance siya sa antibiotic na yon, na hindi pwedeng mangyari na susceptible siya. Well, there are some cases, basta sigurado kayo sa pagka-identify na siya talaga, then baka may mali sa ginawa niyong procedure. Yun lang yon. So that would try now to add in on how you will try now to attack or identify later on the specific genus species of your particular isolate under the family of Enterobacteriaceae and several others you have there. So you have also for Klebsiella erogenes. So you have ampicillin, amoxiclav, ampisul, ticarcillin, and you have cefazolin, cefaltin, and then the rest, cefoxylin, cefotetan. And Morganella, if you try not to look also the area of proteus, okay? For mirabilis, these are susceptible to ampicillin, but it will be vulgaris and pinearii, usually they are resistant to penicillin. We have an argument with my former section head in the micro, having identified it as vulgaris, but I told him that is not vulgaris, but that is mirabilis, just because of this um, susceptibility of ampicillin to the isolate. So you can use this one as well. But again, this is not so of the biochemical test, but this is more on susceptibility testing. So I hope there's a lot, but I hope you have got an idea on how to review it. You may not have to already totally absorb all those things, but I hope it will guide you and try now to review your Enterobacteriaceae. Thank you, Ma'am Dom.